So in that process, like looking back, the first process, chopping up the DNA, you can't do that with a piece of scissors or with a pair of scissors because the DNA is obviously so small. So we're actually doing that with what's called restriction enzymes, a chemical that goes in and breaks apart the DNA at a specific location. So what do scientists do with that? They identify the gene of interest in this section of DNA in the donor species. So you can see in step 1, we've got the identification of the gene of interest. Then they put in a chemical that breaks apart the gene of interest at either end. So basically slices the DNA in 2 spots and pulls out the piece that they want. In this example, you can see the A's and T's and G's and C's. It's cutting between the first A and T and the last A and T, and it's cutting out that little fragment. And then the restriction enzyme binds to the target base pair and cuts the strand of DNA. And so now that piece of DNA, that fragment of DNA that the scientists want to be able to work with, has been chopped out of the long strand of DNA and isolated so that they can use what they want. In the next step is called amplification, where we're going from one little segment of DNA to 2 identical segments of DNA. So what scientists do is they basically add this solution that has isolated segments of DNA. They heat it up so that it separates from double-stranded DNA into 2 single strands. So it's so hot that it's being moved apart. And then the enzyme, which is called DNA polymerase, is added. And it's got all these free bases that are floating around in the solution, and as they cool it, it starts to come back together. And so we go from one strand and the complementary strand on the other side to all those little free bases that are floating around out there. As it cools, those bases attach to the one side, and then they attach to the complementary side, and you have 2 identical pieces of DNA, each of which have gotten those free DNA, base pairs that are being attached to it. So we go from 1 strand of DNA to 2, and then each of those 2 strands of DNA, they do it again until they have 4, and each one of those, the 4, turn into 8. And so as we go along, it says you can go billions of times. We can make huge amounts of this DNA to be able to have what it is that we want in a much larger amount. So the next thing that happens is inserting the DNA using plasmids. So we take this DNA that in this target segment, and we've gotten it all by using the restriction enzymes and amplification. And then scientists make a single cut in what's called a bacterial plasmid. So if you remember back to our single-celled bacteria, they have their DNA in this circular shape, and that's called a plasmid. What scientists do is basically they cut out a chunk of it, and they put the piece of DNA in there. That's the piece that we want to replicate or make more of. And so they fit perfectly together, and they're inside of the bacterial cell. And as the bacterial cell grows and divides into 2, and the 2 divide into 4, and the 4 divide into 8, we've replicated those pieces of DNA that we want every single time that bacterial cell is dividing. So one of the things that we can do is we can create what's called a gene library. So basically lots of genes. You can see in step 1, we've got gene A. We put it into plasmid A, which is the circular DNA that's found inside of that bacteria. We stick the plasmid inside of bacterium A, and we put it all together. We do the next thing for gene B. So we splice it apart, We put it into the plasmid, put the plasmid back into bacterium B, and then it starts to replicate. And so we have all of the different bacterial cells containing all the different fragments of DNA that we want from those original DNA in much larger amounts. And we can create this entire library, if you will, of bacteria that have in it what we expect to find there. So in order to identify these genes, we can use what's called a DNA probe. So we go into the gene library and locate the gene of interest among those millions of clones of gene library in the gene library, and the bacteria are washed with a chemical that breaks it down so it gets rid of that cell wall around the outside of the bacteria and makes the DNA come apart back into that single strand. And then a radioactive probe is washed over all of that DNA. So as it's all out there in the single strands, the DNA probe is put into all those strands that are all pulled apart. And the DNA binds to the probe and glows because it's radioactive. Like, you know, you think of Chernobyl, and sometimes in scary movies, things that are radioactive are glowing. Well, in this case, they are actually glowing. So we've got our radioactive DNA probe, binds to the DNA, and it glows under this UV light because of radioactivity. It allows us to easily identify that gene. So if you're going to take something out of this portion here, the methods rely on naturally occurring restriction enzymes for cutting DNA apart. So we're looking at something, taking something that is natural, and we amplify the DNA, insert it into bacteria or viral vectors, and go through the cloning process, making lots more of it, and then finally identifying the cells that have the DNA of interest inside of it. So it sounds like this amazing process, and it's amazing technology that scientists have been able to figure out, taking something that occurs naturally and using it as technology in order to do something good. So here you can see we have biotechnology improving our food. It used to be that corn didn't look anything like those bright yellow kernels of corn that we think of as corn now. Back before we started agriculture and messing with corn, ancestral corn, if you've ever seen during Thanksgiving time here in Ohio, sometimes they pull out the multicolored stalks of corn that have like the purples and the greens, and because we have bioengineered corn through modern agriculture and now through technology venues, we're able to make food that's more nutritious and that makes farming more efficient and more eco-friendly. Some people say, oh, this is amazing. We're getting more food so we can feed more people here on Earth. It can help to combat starvation, combat nutritional deficiencies. But other people think genetically modified organisms are unnatural and that we're not meant to eat it. And so if you don't eat organically or things that have not been bioengineered, then that could be harmful to your health. And so there's a lot of controversy out there about this right now. So one of the most genetically modified plants is rice. And if you think about all of the people over in China who have rice as a staple in their diet, maybe 500 million of those people are malnourished. If we could genetically modify rice in order to contain more vitamin A, that is nutrient-rich golden rice, it could help to prevent deformity, it could help to prevent malnutrition, it could help to make people healthier. And so that is, I think, a good thing. So the way that golden rice is produced is that scientists have identified a gene from daffodils that makes the beta-carotene production, and they've taken that gene and they've placed it into bacteria, grown more and more and more of it, and then used biotechnology to pull that gene out and modify white rice into golden rice that has the beta-carotene in it. And it says that almost 10% of the world's people suffer from vitamin A deficiencies. Oh, there's my dog. Hold this for a second.